Good evening, everyone. My name is Himanshi Singh. Um, I'm a PMRF PhD candidate in BSB Department of IIT Kanpur, and I'll be taking an extra session on animal physiology by Professor Manik Das. As you might have known, that we have covered all the week's lecture in the last week, all the week 12 lectures and live discussions and all the assignment questions. I hope that you guys had fun learning this course, attending this course, and attending all the live sessions which uh, the course team members were taking. This extra session is just for basic information. It has nothing to do with the exam or the coursework or the assignments anymore. So this, I wanted to take this extra session to for you guys to understand a very small thing which we might have missed. I'm not saying we have missed exactly, but we haven't talked about it much. If you guys remember how we started the animal physiology course, we started with integumentary system, and if not that system, before that, in the first week, first lecture, we discussed about how we can study animals, right? There were different hierarchy at which we should understand things, and that is how we'll get the holistic approach and holistic learning about our system. Correct. So uh talking about that, we started with certain uh systems: integumentary system, skeletal system, muscular system, neuronal system, and all those systems. But we haven't thought of what exactly or who exactly is doing these things, right? Correct. We studied about, for example, muscular system. So in muscular system, we studied about different types of muscles, how they interact with the neuron, how they act when the stimulus is there, what is the contraction relaxation process, how the nerve impulse is transferred from from the nervous system to the skeletal system, so that we make the movement at the end of the day, right? Because that is our action against the stimuli or the stimulus which we got from the surrounding. But who exactly sensed it? So if we talk about that, we will say maybe our sensory organs, right? We have different sensory organs, correct? Eyes, nose, ears, skin, all those things. So if I ask you this, who sensed the change in the environment or the stimulus? You will say our sensory system. It was the one which was communicating from like communicating the information of outside world to our inside system, correct? Now, even in the sensory system, what exactly is sensing things? Then you will say there are many uh, receptors available, right? Many proteins which are available, or many small molecules which are available, which changes, or ions at the end of the day, right? Which changes either in concentration or charge or in polarity or all those things. If you guys can compile all the information which you have studied throughout the course and put these variables here, we'll say receptors probably, right? But the receptors are present on what? Then you guys will say on the membrane, correct? On the cell membrane. So then we stopped our discussion at this point. How the stimulus is being sensed from outside world to the inside system. We stopped at here. Like, these are the sensory organs. These are the elements involved in sensing. So that's it. But these sensory organs or these receptors or these things are present on cell membrane. And we have discussed, I personally covered cell membrane quite extent in one of the week's lecture when these sensory mechanism was being introduced in the course session, right? So that cell membrane is membrane of what? It is a membrane of cell, right? So at the end of the day, everything is happening where? It is happening at the cellular level. So if you remember the hierarchy in which we can study animals or study living system was based on like macro properties, then at cell level, and then at small molecule or biomolecule level, and then at the end of the day small molecules like uh, ions per se, right? Ions, ATPs, all these are small molecules. So everything is happening inside the cell, and obviously you guys know that cell clumps together to form a organ, right? Different organs come together to make different organ systems come together to make the organism, right? So we have studied about the organism from the macroscopic point of view, what we are seeing, what is happening at a macroscopic level, what changes we can see via our naked eyes. We dig sometimes into the molecular aspects also, but not in what those molecular actions are being taken place, correct? So I figured it out that it is important for you guys to know some basic things which we are right now, which we are studying about, right? We are studying about animal physiology. So animal physiology means the study of animals and their behavior and their systems. And as I just mentioned, systems are made up of different organs, correct? Excuse me, different organ systems. Different organ systems are made up of different organs. Different organs are made up of cells or tissue for that sake, right? After this, when we have our tissue, and tissues are made from cells, so we should know and we should have some very clear understanding about cells, correct? So for our extra session today, instead of dealing with a particular system, because we have already covered those things in the 12-week extensive course, and you guys have some brief idea about most of the systems which are available, so you can always pick your site, pick your system, pick your interest, and just dig more and more into it. I only tried to give you some additional information which was like adjunct to the information which were being provided in the sessions, okay, or in the course lectures, so that you might get intrigued by some system and you can go and further read more about it. So with this background, I would like to start today's discussion with cell. I hope that every one of you know what is a eukaryotic cell or a prokaryotic cell. At the end of the day, a cell. I've kept this very, very, very minimalistic view of a cell. We know much more about a cell now in comparison to what is being depicted here. But this is just a representative way of saying that there are two different types of cells. And right now, as I've discussed, why it is important to study cell. But at the end of the day, every single thing is coming to the cell. The cell is taking the action and things are getting multiplied and multiplied and multiplied to the organ system level. And at the end of the day, then the organism is taking that action. Correct? In every system, whatever systems you have studied till now, the site of action or the site of uh, changes or communication, it happens inside the cell, right? So there are two major different types of cells we can categorize: eukaryotic cell and prokaryotic cell. Okay, so in eukaryotic cell, there are certain features which are common to both eukaryotes as well as prokaryotes. I'm not digging much into eukaryotes and prokaryotes. I want you guys to have a very clear understanding of that this is a eukaryotic cell and this is a prokaryotic cell. But at the end of the day, they both are cells, right? You might think that it's a very rudimentary information, but trust me, there are many people who don't get the concept of a prokaryotic cell and a eukaryotic cell. And here I'm not saying that you know the differences between these two cells. Everyone can see online or in any standardized textbook, you will get to know the differences between a eukaryotic cell and a prokaryotic cell. What I'm inferring to in understanding about the eukaryotic and prokaryotic cell, for example, you know that eukaryotic cell is more complex in comparison to a prokaryotic cell, obviously, right? But all the functions which are being performed by a eukaryotic cell has been present to some extent, not, I'm not saying at equivalent rate in both of these cells, but most of the functions are also being performed by the prokaryotic cell. So a question should arise in your mind that if a simplistic system is available, and here simplistic system means prokaryotic cell, right? So if a simplistic system is available, why or what was the need of generating a complex system? Correct? We always want to be in the like the how much we can simplify. You can see this principle working in every area of your life. If you want to understand something, you want to understand means you want to simplify it, that you are able to understand in a simplified terms, and hence you can make other people others also understand the concept. If we talk about food, right, so we make food very complex. We add so many things into it, but at the end of the day, as we have already understood some basic flow of digestive system, you know, even the complex molecules are being, at the end of the day, are being converted to simpler molecules, because what we want from the food is the energy, and energy can only be extracted when the simple molecules are present. Okay. So if the universe, and here the universe, I mean most of the principles which we see, they work in simplifying things, not making it complex. So why the evolution has been so opposite to the universal things, that from a simplistic cell, a complex cell was made, and those complex cells are the one which are kind of dominant, but we cannot say that they are the dominant, but still we have more complex cells. So knowing only the differences between a prokaryotic and eukaryote, it's a very textbookish knowledge. What I want you guys to imagine and try to answer this question, that if a simple cell is able to do all the functions, correct, then why do we need a complex system? A similar example can be, a similar analogy can be made. For example, I don't know how many of you are aware about the evolution of hemoglobin in our system, right? We have studied the circulatory system. We know that we have RBCs. RBCs have a hemoglobin which has the oxygen carrying capacity, correct? And this is the only pigment which allow us to breathe in air, extract oxygen from it, circulate, provide it to the cells, do the metabolic work, extract the energy, and then we exhale out the CO2, correct? And hence it's a very important molecule for our survival. Uh, yeah, obviously for all the aerobic organisms which has this pigment, not necessarily hemoglobin, but any pigment which has the oxygen carrying capacity is critical for survival, right? It is an absolute need for the survival. So if you go to the background of hemoglobin and look at the evolution of hemoglobin gene or the hemoglobin protein itself, you'll come to know that even in our development itself, like we humans, when the pregnancy has been achieved and the organ and the fetus is being developed, then there are different stages of hemoglobin itself. So there is one stage of hemoglobin in the fetus which is called hemoglobin. It is a hemoglobin like we all adults have right now, but it has some differences, because of which it is called hemoglobin. So we are not gonna go dig deep into it, but I just want you guys to know that there are two different types of hemoglobins. Okay, one is the adult hemoglobin, which all of us have it right now, and one is the fetal hemoglobin. It is the hemoglobin which, when we were fetus, when we were inside the womb, at that time those that type of hemoglobin was expressed, and our body was filled with fetal hemoglobin, fetus hemoglobin. Okay, So my point is, when I'm trying to make parallels with eukaryotic, prokaryotic, meaning simpler and complex system, and the analogy is the fetal hemoglobin and the adult hemoglobin. So what I want you guys to understand with respect to the fetal hemoglobin and the adult hemoglobin is that fetal hemoglobin is more efficient in carrying oxygen. Meaning, for example, right now we all are adults here. We all have adult hemoglobin gene expression. Probably with a high probability, I can say that most of us have adult hemoglobin. Okay, there are exceptions also, but we are not considering those things right now. So we all have adult hemoglobin, and I'm saying fetal hemoglobin is the one which has more efficiency. So making it more simpler, what I am saying is that if you take a equal amount of fetal hemoglobin and adult hemoglobin, fetal hemoglobin is able to carry more oxygen per unit volume or per unit weight or whatever unit you want to put it in, per unit in comparison to the adult hemoglobin. Correct? So a question should come to your mind that if it is more efficient, if fetal hemoglobin is more efficient, why it is being turned off, and adult hemoglobin, which is less efficient in comparison to the fetal hemoglobin, is being turned on? Correct? It's a very basic question. If we already have something very efficient in our system, why it is not being carrying forward throughout our life? We want more efficiency, right? And here by efficiency, I mean if you have hemoglobin, if you have more hemoglobin, which means you have more oxygen carrying capacity, which means you have more stamina, right? So all the athletes and all the trekkers and all the players around the world who are involved in a lot of uh, cardiac and aerobic work, they have in general more hemoglobin content, because they need more oxygen for the amount of work which they are involved in. We all want that. Then why fetal hemoglobin is not being carried forward throughout our adult life? Correct? Talking about prokaryote and eukaryote. So if prokaryote is a simpler system, it is able to perform all the functions which a eukaryotic cell, most of the functions which a eukaryotic cell perform, then why there was a need for a eukaryotic cell, right? So understanding the differences is a different thing, but we should understand 1 minute. Yeah, Karthik, correct. But my question is why we should not have fetal hemoglobin throughout our adult life. There are answers out there. There are different speculations out there, but we are not gonna discuss much on it. I'm just putting a context in every one of you, that knowing the differences is a very textbookish information, but we should understand the concept and the logic, because even in physics and chemistry we understand certain principle, we study about certain principles and universal laws and those things. All those rules and laws should apply in the biological system also, because we are also part of the universe, right? We are not outside the universe, we are not away from the universe, we are part of the universe. If there are certain rules which are being followed for the universe, those rules will be applicable for the biological system also. So it's like an interdisciplinary understanding. If you know the laws of motion, right, for uh, uh, bigger bodies, smaller bodies, those same laws of motion apply to our biological system, cells, vessels, blood, all those things. Same things will be applied, right? That is why we study science as a whole subject, not we separate science at a very later stage in our education system into physics, chemistry, bio. In general, we study science as a whole subject, right? Because everything is connected. So with respect to eukaryote, prokaryote, you all guys are very smart. You must know what are the differences between eukaryotes and prokaryotes. But I want you from now onwards to think what was the need of having a complex system. Correct? And gradually we will come to know, if we talk about organelles, so there are different functions which are dedicated for those organelles, which earlier was being performed by the cell or a prokaryotic cell in this case, but it was at very less efficient. It was not an efficient way of doing that, right? So our system always makes a trade-off. And here our system means our biological system, living beings in general. You can see this phenomena from macroscopic point of view as well as to the microscopic point of view. If we talk about macroscopic point of view, so okay, let me give this example. You must have seen many predators chasing their prey, right? But if the prey is, for example, too fast or too far, or something is too much, that you need to calculate whether you want to input that energy in getting that prey or you want to leave it. So here the trade-off will be, you need to compare after getting the prey, the amount of energy which you are going to get from the prey, is it equal to or more than the amount of energy which you have put in to get that prey. I hope I'm clear. I'll try to make it more simpler. What I'm saying here is that in the living world, there are always trade-offs. If you want to have food, but the amount of energy which is required to get that food is more in comparison to the energy which you will get after getting that prey, then it's not a good deal, right? Because you have uh, given a lot of energy in comparison to what you are getting out of it. So your energy balance is getting lower, it's getting into negative, correct? This is the reason that in the biological system, most of the decisions are based on trade-offs. Okay. Now, this was an example of a macroscopic system. Now I'm talking about microscopic system. What information we lose when a simpler system, which is okay, it doesn't require much energy, it is working quite well. But what if we make this expenditure of energy and make the system a little bit more complex? So the amount of work per unit time and the efficiency which we are getting by inputting this amount of energy and making the system complex is more in comparison to when the system was simpler. The organism, the cell, the evolution has taken it into consideration and has put that energy into it. I hope that I'm not confusing you guys. It's a basic trade-off, and even we do this, right? If we talk about trading, or if we talk about anything which we are making, for example, you guys are present here today, so you wanted to learn something and you are leaving something, maybe some festivities are going on or something else, or you had something planned earlier also for a Saturday evening, but you guys are here, so you have made a trade-off because getting information or particular knowledge was more in comparison to the energy or the things which you were getting when, if you have taken the other path. So it's a trade-off. Similar things are being performed by our cells unknowingly. It's not unknowingly, it's how evolution has progressed. That we always want to have the least energy system because that is the most stable system. We want to spend least energy. We want to gain maximum benefit of a thing. And here you can put anything. If we talk about these three aspects with respect to cell, a cell will always want to be in its lowest energy state. It doesn't want to work until and unless absolutely necessary. And here necessary means that the cell needs to spend energy because the amount of energy you're going to get, or the amount of thing, or the amount of anything here, just put that this aspect will change based upon what phenomena we are talking about. Okay, so a cell knows this, that if I spend right now 20 units of energy, I know that definitely I'm going to get 50 units of energy. The net gain is 30 units of energy. Hence, the cell will spend this 20 units of energy. It won't conserve it because it knows that it is going, after spending 20 units of energy, the net profit is going to be of 30 units energy. So the system energy or the system commodity or the system unit will increase. Correct? So there are always trade-offs being carried out, both at macroscopic level as well as at microscopic level. Correct? I hope that with this new perspective of thing, that we always target for least energy system, we always target for simplistic system, we always target for less complex system, because that is how it is easier for our system or our things or our surrounding to work at maximum. With this point of view, now try to look for the differences between a prokaryotic cell and a eukaryotic cell. You will get some very different point of view, that why something which was present in the prokaryote has been carried to the eukaryote, or some things which were not present in the prokaryote have been evolved in the eukaryotic cell. So you will get the relation, that why this property or this function was needed to be more efficient, though it is more energy consuming or it has more steps, but still overall it is a profit, it's a profitable change. Okay, so that was the thing with respect to a prokaryotic cell perspective and a eukaryotic cell perspective. Now just 1 minute. Okay, I can see many of you here. Do you guys want to say something, or just give your brief introduction? If you guys are interested, just unmute yourself and say hi from where you are and all those stuff. We can, today's the last class for this course, so let's know each other if you guys are okay with it. Anyone, if you want to unmute, just raise your hands. Okay, cool. No problem. If you guys are not comfortable, we'll move ahead with our discussion. Yeah. So as I mentioned, that it was the very simplistic representation of a cell in the previous slide, this one, right? I hope that you might have observed this a lot, and you know, and in general, it's a very basic information for you guys, especially for our biology people. Here, this cell which you see is the one which is kind of more detailed from the previous figure. Obviously, it has covered most of the things which are present in both, which are present in different cell types. But I'll still say that this is not a complete picture. We are still figuring out new things about cell. With the advent of technology, the more the more high resolution techniques are developed, the more we will come to know about our cell. I hope that you guys are aware that in recent years, I guess within 2 years or maybe this year itself, a new organelle is being found. Apart from the organelles which you might have studied in your 10th or 12th, now there are new organelles also. So how come since so long, like in 1850s, if I'm not correct, or 1805, the cell was 1605, the cell was discovered. I might be wrong here, so don't quote me. So it's been a long since we know that cell exists and it is the functional unit of life and all those information. But how come still we are discovering new things about it, right? So this happens gradually and parallelly with the advent of technology also. So for the cell to know that the cell exists, the technology, that is the microscope, needs to be developed first, right? Then only we'll be able to see microscopic entities like cell. Similarly, now if you want to see inside the cell, we need more high resolution techniques which can actually look inside the cell, right? After that, the technology should move forward more in increasing the resolution as well as the specificity of different things inside the cell. Then only we'll come to know about different organelles which are present there, right? So all the fields are interconnected, especially in science. So if you're developing a tool, or if you're working on a technique which, for example, can distinguish L form or D form of an amino acid, so it will be completely same. Only one functional group will be on the right side and one functional group will be on the left side, right? Then you can say there are two different forms of amino acid. But if you don't have any such technique, you'll say it's a amino acid only. So I hope that this is clear. If you want to study one thing, the technology, not necessarily in your own field, in other fields, the technology should be in par with the current development of your field. Okay, so this is one of the detailed representation of a cell. It has most of the information which we are aware about cell, but I'll still say that many things are lagging here, and many more things will come in future which will completely change the image which we are seeing today for a cell. Okay, so if we look here, that ideas about cell structure have changed considerably over the years, right? Early biologists saw cells as simple membranous sacs containing fluid and a few floating particles. Today's biologists know that cells are infinitely more complex than this. So you can get the beauty of this sentence, right? So why people thought, or early biologists thought, that cell is just a membranous structure with some fluid and some particles floating in it. Why? Because the technology which was available at that time, the microscope, was only able to show them this, that's okay, something round is there, which means they are separated, which means it's a membrane, because it's a flexible thing going on, it is moving. If you look at the E. coli, then they are changing their shape, they are undulating. Amoeba, look at the amoeba, right? So they just saw there's something membranous and fluid is there and some particles are there. But today we know that those particles are not just the particles, right? They are the proteins, they are the organs, they are different things. Now we are aware of it. So with the advent and with the advancement of the technology, especially the spectroscopic techniques, we will be able to like unmask the cell, hopefully one day with 100% precision. And that will be a really really great day for biologists and in general everyone, because we will be knowing everything about the functional unit of life, right? That is more important. We don't know how we work completely, then how we will be able to solve things. And even if that is not the motivation, that you want to solve or resolve thing, it's a good curiosity to have, right? How you function, how you behave, how you change based upon the surrounding conditions or based on the stimuli. So to know that, if you know the simpler unit one, if you know the cell, you can predict the behavior, or you can expect certain things to happen. Then you can apply this knowledge and information to different fields of life. So if you're aware of deep neural networks, right? Deep neural networks is a kind of, for example, machine learning technique or machine learning algorithm, which was taken from how the neurons are connected in our brain, or how they function. Not exactly the replica of it, but obviously they have taken hints. Okay, one neuron has multiple dendrites, only one axon. It transfers the information from one. It takes input from multiple things, then transfers it to the next input, which has like dendrites for different axons. So similarly, in this deep neural networks or neural networks for say, this is what happens, right? You get input from different files, or you get different inputs to one node. Then the information is being processed, and that information is being fed to the next link, which again is receiving input from multiple inputs. So knowledge or information of one field has been used to make something in different field. Hence overall understanding of things, especially living system, is in itself is a very intriguing quest to follow. Okay. So, the cell is the basic structural and functional unit of all forms of life. Obviously, we know this. Every cell consists of cytoplasm enclosed within a membrane and contains many macromolecules such as proteins, DNA, and RNA, as well as many small molecules of nutrients and metabolites. The term comes from the Latin word cellula, meaning small room. Okay. Now, if we want to dig into protein, DNA, RNA, most of you are aware of proteins, DNA, and RNA. I'm sure that you guys have heard about these things. You guys might have more information in on these in comparison to me. But again, I want you guys to put your attention into why do we actually need so many molecules, right? If at the end of the day, protein is the one which has to function, then why not have proteins since we are like, since the zygote is formed or since the life has begun? Why do we need DNA? You might have an answer for this because we need to store the information. Proteins are big and all those generic answers. Then my question will come, why do we need RNA? If information is stored in DNA, DNA is the one which is being read, what is the need of RNA? Correct? It's a legitimate question. Anyone of you want to give it a try, that why do we actually need RNA in our system? It is just an intermediate, right? DNA is the one which holds the information. Protein is the one which performs the function. So just have both of them. Why to have an intermediary step? Again, it's a complex system. Two-part system is okay. I don't know what is happening. I'm sorry about that. Yeah. So yeah. So two-part system is simpler in comparison to three-part system, right? So we could have a DNA which can be directly converting its information to protein, because at the end of the day we want protein. So why do we need to have a RNA in the system? Anyone of you want to give it a try? In the chat box also you can write if you want, no problem at all. But just let me know that you guys want to attempt it. Any even wrong answers are welcome. It's a very open question. Why do we need RNA? We study about DNA, we study about RNA, we study about protein. But why we have these things? Why do we have these things? Protein, I have justification. DNA, I have justification. What about RNA? So anyone wanna take a guess that why do we actually need RNA in our system? Kindly drop your answer in the chat box. So you, again, I'll repeat myself. You guys have 1 minute. It's not a question. It's nothing at all. Just whatever is coming to your mind, just try to pin it down, because that's how you'll develop thinking ability. And that is more important than just mugging up the information. Trust me. So please do write. Why do you think, the question is, why do you think that we need RNA in our system? RNA helps in transferring amino acids for coding. It carries information from nucleus to cytoplasm and helps making proteins and helps synthesis of protein. For coding and synthesis of protein. It helps, but why DNA needs help? Why it needs help? What will go wrong if we don't have RNA? What will go wrong? At the end of the day, the DNA is the one which is being transcribed, right? After transcription, translation happens. I am saying stop the transcription process, directly have a translation process. Make protein directly from the DNA. Don't put any intermediate. In case of some virus, RNA is the. I agree. So we have such kind of systems, right? I'm not talking completely bizarrely that, oh, I'm completely eliminating the RNA. Systems are there. They have like some, they have figured out ways in surviving with these exceptions also. So you guys need to think, what is the role of RNA? Whatever you are telling me, it is the function of RNA. Okay, whatever. DNA has evolved from RNA. If we go with this, then why do we need DNA? The question will come, why do we need DNA then? If DNA has evolved from RNA, RNA was already there. RNA was functioning properly. RNA is the one which makes protein. So from RNA to protein is a one-step process. From DNA to protein, it's a two-step process. Which process is simpler? One-step process. Then why we went back to two-step process? That question will arise. You getting my point? The loops, the circle which we are talking about. DNA to protein is a two-step process because DNA first needs to get converted into RNA, then RNA will get converted into protein. While making protein, at the end of the day, everything is making protein, right? Protein is the one which is the functional form of a change, correct? From RNA to protein is a one-step process. There is no intermediate involved in making protein from RNA, and such systems exist. Then what was the need of DNA having intermediate RNA and converting the DNA information into protein? DNA is more stable as compared to RNA. See, these all are the answers which we have been learning since uh, our school days. I agree with all those things which you guys are saying. Those are not wrong at all. Those are not wrong. This is what everything comes down to. More stability and everything, and all those things which you guys are saying. But what I want you guys to do is that push yourself out of these answers. DNA is more stable in comparison to RNA. Why do we need a more stable system when the earlier system was already performing well? Why do we need DNA? Okay, I'll give you more information and I hope that you might understand what I'm trying to imply here. Let's take some common example. Okay, for example, you have one notebook. Okay, you have one book and you want to send the copies of this book, or you want to have this book in all of your rooms. You have, for example, you have 3 rooms at your place. You have one book and you want to have this book in all the 3 rooms simultaneously. How is it possible? How can you make this happen? uh, we need more stability to reduce inaccuracies and mutations. If you're going there, why you need to reduce inaccuracies and mutations? We want our system to be mutated, right? Then only the variations will come, then only the adaptation will come, and then only the evolution will take its part. Correct? That can also be considered in this way. Yeah. So what I'm saying now, answer for that. I'm saying you have 3 rooms at your place. You have one book. You want this book to be present in all the 3 rooms simultaneously. What will you do? Anyone? DNA replication made possible by. I'm not talking about DNA at all. Right now, the question is about I have one book, I have 3 rooms, I want this book in every room. What I have to do to achieve this goal? Divided into 3 pieces. Great. Now, if I divide this book into 3 pieces, for example, part A, part B, part C. So in room 1, I have part A. In room B, I have part B. In room 3, I have part C. But my question was, I have one book and I want this book in all the rooms. The suggestion which you gave me, it is having a part of my book in room 1, a part of my book in room 2, a part of my book in room 3. My whole book is not anywhere. It's not present at all. The book is changed now. 3 copies of book. Great. Now what I have to do? I have to photocopy it. Easy solution. I'll make Xerox of my book, right? And I'll keep it in room 1, room 2, and room 3. Everyone agrees with this? Correct? Now put the same information with respect to our cells. Each and every cell of us has all the information and the chromosomes which is available in your germinal cells. Do you guys agree? Apart from XY chromosome, obviously it's there, but not expressed. It's the same thing. But all the information, if you take out your skin cell and sequence its genome, if you take out, for example, your liver cell and sequence out its genome, they will all have the same chromosome pattern, correct? Indicating that room 1 has the same copy, room 2 has the same copy, room 3 has the same copy, correct? Here comes the role of RNA. Now, the first suggestion which Karthik gave is to divide it into 3 pieces. Correct? If you're dividing a book into 3 pieces, here the book is the chromosome or the DNA which we are talking about. If you divide it into multiple pieces so that part of it will be in different cells, it's not an effective way. Because what if in future my say if an injury occurred in my foot, but in my foot the cells which are present, they don't have wound healing genes, right? Or the genes involved in wound healing, they don't have it, then indicating that my foot wound will never heal because that part of the gene is not present there. To resolve this, what we did, we made copies of our DNA. How we make the copies of our DNA? Via RNA. So we have a master copy, we have a DNA in every cell, stored in every cell. Correct? Now we take out the chapters, one chapter at a time, depending upon the need or depending upon the chapter which we want to read. If a wound has occurred in my foot, the genes or the chapter which is associated with wound healing, it will get translated via RNA and it will help in wound healing. What if my wound is in my hand, right? And the wound healing chapter is in my foot. Will it help me? No. Hence, the RNA role comes here. The master copy remains conserved. To take out chapters, to read different chapters, at different time, photocopies of those chapters are performed in different parts of the cell depending upon the need. Correct? I hope that I made some sense that why we need RNA. RNA is just a mediocre thing which helps in associating and making sure that a master copy is remained as it is, because you never know in future what need you might what you might need in future. And the moment you need something, a photocopier comes. That photocopier is in the form of RNA, that chapter is in the form of RNA, and that RNA is being translated into protein and the function is being performed. So I hope that you now understand what I meant by a more complex system and a more simpler system. Definitely having a RNA is a complex system. It's an energy expenditure system. But the efficiency you are getting and the overall survival, help in the survival you are getting by spending this amount of energy is more in comparison to spending this energy right here, right now. Okay. Okay, let me go to the chat box. Can we say RNA helps in producing identical proteins? It depends upon the RNA also. So if you are familiar with the molecular biology aspects, a single RNA can give rise to different proteins also. So identical proteins we cannot say. Okay. Moving forward. So as I mentioned earlier, both eukaryotic and prokaryotic cells have some common features, and there are certain features which distinguishes them. Obviously, that is why there are two different types of cells. So if you look at the this little bit more detailed image of a cell, we can start from here. Okay. So what is the first thing which you are seeing here is the centriole. We'll see in the coming slides what centriole is. So what are the parts of a cell basically? So first one is the centriole, then centrosome, then lysosome, peroxisomes, rough endoplasmic reticulum, cytoplasm. Then we have secretory vesicles. We have cell membrane or plasma membrane. We have Golgi complex or Golgi apparatus. We have smooth endoplasmic reticulum. Then we have nucleus. Further, nucleus has many parts such as nucleoplasm, nucleolus, nuclear pore, and nuclear envelope. I hope that every one of you can see my cursor on the screen. And if not, please let me know in the chat box. I'll switch it to laser pointer. Okay. So what does nucleus have? Nucleus has nucleoplasm, nucleolus, nuclear pore, and nuclear envelope. Okay. The next is the ribosomes, then mitochondria, then microvillus, cilium. So these are some of the things. This is not one particular type of cell. It is a generic representation of a cell. So if we move from outside to inside, so what will be the first thing which we will encounter when visualizing a cell? As you can see, what was the earlier biologist saw? It was a membranous structure filled with some fluid. So what is that membranous structure? That membranous structure is the cell membrane. Okay. So every cell in the body is enclosed by a cell membrane. The cell membrane separates the material outside the cell, extracellular, from the material inside the cell or intracellular. It maintains the integrity of a cell and controls passage of materials into and out of the cell. All materials within a cell must have access to the cell membrane or the cell's boundary for the needed exchange. The cell membrane is a double layer of phospholipid molecules. Protein in the cell membrane provide structural support, form channels for passage of materials, act as receptor sites, function as carrier molecules, and provide identification markers. So if you guys are following the previous sessions and lectures as well, you know that we have discussed about cell membrane to quite extensive details, right? And in that, we have also discussed about the role of different proteins in it. We have also discussed about biomarkers, right? We have also discussed about the need for communication between the inside of a cell to the outside of the cell. So everything which is written here with respect to cell membrane, we have actually discussed. So feel free to ask any question if you have, and I'll be happy to answer if I have that information, because cell membrane is a thing which is common to both prokaryotes and eukaryotes, right? Both prokaryotic cell and a eukaryotic cell has plasma membrane, though depending upon the organism and even to which domain of life we are talking about, how they are made what they contain might change, but their basic role and functionality stays the same. Okay. So next, talking about the cell's nucleus, because this is the one which is again the brain of the cell. And why it is called brain? Because it has DNA, right? It has DNA, and hence, if I want to perform some action, I need protein, as we have quite established earlier. Those protein will manifest themselves as a change in stimulus, whatever we are receiving from the outside. Okay. So cell nucleus, if we like read a little bit about it, a cell's information center. The cell nucleus is the most conspicuous organelle found in a eukaryotic cell. It houses the cell's chromosomes and is the place where almost all DNA replication and RNA synthesis occurs. The nucleus is spherical and separated from the cytoplasm by a double membrane called the nuclear envelope. Space between these two membranes is called perinuclear space. The nuclear envelope isolates and protects a cell's DNA from various molecules that could accidentally damage its structure or interfere with its processing. During processing, DNA is transcribed or copied into a special RNA called messenger RNA. This mRNA is then transported out of the nucleus, where it is translated into a specific protein molecule. The nucleolus is a specialized region within the nucleus where ribosome subunits are assembled. In prokaryotes, DNA processing takes place in the cytoplasm. So here you will get the idea of when I mentioned earlier that why do we actually need RNA? And as mentioned, it's a copying mechanism. We need to copy it and all the story which I have made. So you should ask, why do we need to make a copy? So the answer lies here because DNA is deep inside the nucleus, right? And why it is inside the nucleus? Because it needs to be protected from the cytoplasm. Why? Because in cytoplasm, most of the metabolic activities are taking place. And each and every instant, there are thousands and thousands of biochemical processes which are taking place inside the cell, indicating that there will be multiple small molecules or changes in the pH or temperature, localized pH or oxygen availability, those things which might damage DNA. And we cannot afford our DNA to be damaged, right? Hence, to preserve our DNA, we kept it inside the nucleus. But now the problem is our site of action is cytoplasm. From cytoplasm to the outside world, or the extracellular matrix, from extracellular matrix to our movement system, right? So here the trade-off will come. So to protect our DNA, which was more important, we made a system that it can be copied to a RNA, to copied into some entity, and that entity can go out. If that entity is being damaged, we can still somehow surpass this process. But if our DNA is damaged, we cannot surpass this process, right? So a trade-off is being made here also. So you can actually see this throughout the living system, that at every stage, many trade-offs are being carried out. Okay. Next is the cytoplasm. The cytoplasm is the gel-like fluid inside the cell. It is the medium for chemical reaction. It provides a platform upon which the organelles can operate within the cell. All of the functions for cell expansion, growth, and replication are carried out in the cytoplasm of a cell. Within the cytoplasm, materials move by diffusion, a physical process that can work only for short distances, right? So what is cytoplasm? Cytoplasm is the fluid which early biologists saw, right? That it's a membranous structure and some fluid with some particles on it. So our cytoplasm is the fluid where most of the biochemical reactions are taking place. It provides us and other organelles a space where they can operate at proper distance. So the representative image, if you see, uh, we cannot see here exactly. Maybe I have other figures and we'll see there. You won't see this much spacing in them. No, a cell is very small, right? And these many things are packed in that small cell, indicating that they are in very close proximity to each other. Hence, the cytoplasm is the place where organelles, reactions, everything is happening at a very compact place. So for a compact place, for a small place, do we need active processes to take place in transferring one molecule to another molecule from one place to another place? Right? This is also one of the reason why the process is so much streamlined. If you go through a process for the synthesis, from replication to transcription to translation to secretion, you will see that everything lies in a same plane, because DNA is present in the nucleus. Nucleus will the DNA will replicate. There the transcription will occur, the RNA will come, it will the ribosome will sit on the RNA. Those ribosomes are sitting on the RNA at the endoplasmic reticulum, which is directly connected to the nuclear envelope. From nucleus to rough endoplasmic reticulum, from rough endoplasmic reticulum to Golgi apparatus, which is again connected to the rough endoplasmic reticulum, from Golgi apparatus to the cell membrane. So to streamline the process, all organelles are situated and arrange themselves in such a way that least energy is being imparted for these processes. Okay, so a cell is actually a very, very, very great example of how to optimize multiple things simultaneously without much loss of energy. Okay, moving to the next part, cytoplasmic organelles. So cytoplasmic organelles are little organs that are suspended in the cytoplasm of the cell. Each type of organelle has a definite structure and a specific role in the function of the cell. Example of cytoplasmic organelles are mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, and many more. These are some major organelles. Excuse me, just give me 1 minute. Sorry for the disturbance. Yeah. So these are some of the organelles to name. There are many, many, many organelles which are available in the cell, which perform different functions at different time. Okay. Some of the major organelles I'm going to discuss. And if you have any queries, feel free to drop in the chat box. So a mitochondria and chloroplast. First of all, do we have chloroplast? It's a very generic question. So do we have chloroplast? Excuse me. Okay. Yeah, obviously we don't have chloroplast. But do mitochondria and chloroplast share some similarities? Yes or no? Yes. So isn't this interesting that an organelle which is not present in us, it is actually present in plants, shares close resemblance with the organelle which is present in us? We say that we are very different from plants or from other species, right? Even 0.1% change in the genome is make different species, right? We are 99.98% similar to chimps, I guess. So it is literally 0.02% change in the genome, which make us humans, and chimps, right? So we are so different with the 0.02% variation. But isn't this intriguing that a similar organ is present in plants, which is not at all related to humans in some way. It's a complete different domain of life, right? This is intriguing, correct? Indicating that evolution, or say how we have evolved throughout the years, have taken some standardized protocols to make the process smooth, right? Because how many permutation and combinations you can have with the limited number of molecules? The molecules are same. We have DNA, we have RNA. In DNA, we have ATGC. In RNA, we have AUGC. We have 20 essential. We have 20 proteins, 22 proteins. Save for that. Then we have different commodities. So we have limited number of molecules, and we have so much variety of it. So obviously there will be chances when you will see certain similarities, and most of the time dissimilarities, but still some similarities of these combinations. So this is very, very intriguing and interesting. Okay. So what are mitochondria and chloroplast? They generate energy for the cell. Mitochondria are self-replicating double membrane bound organelles that occur in various numbers, shapes, and sizes in the cytoplasm of all eukaryotic cells. Respiration occurs in the cell mitochondria, which generate the cell's energy by oxidative phosphorylation, using oxygen to release energy stored in cellular nutrients, typically pertaining to glucose, to generate ATP, aerobic respiration. Mitochondria multiply by binary fission, like prokaryotes. Chloroplasts can only be found in plants and algae, and they capture the sun's energy to make carbohydrate through photosynthesis. I think it is self-explanatory, and you can see the structure of mitochondria and the chloroplast. This black and white part is the TEM image, or the transmission electron microscopic image of the representative structure of the mitochondria and the representative structure of the chloroplast.
मोबाइल दे तेरे को देना
Karthik, can you please like mute yourself? Thank you. And this is the staining image of a mitochondria. You can clearly see what I want when I was discussing earlier about the this density and the cytoplasm part, that this shows that everything is spaced very nicely and neatly inside a cell. No, it's not like this. This is a section of very small section of a cell, and you can clearly see that there are so many pigments or droplets associated here, right, within one mitochondria. So the other organelle will come right here, the other will come here, and all those things are interconnected. They are communicating with each other via small molecule. So it's a very compact place. So you should always keep this in mind whenever you are discussing about some process or something. You should always consider about the association of this particular organelle with the other organelles which might be in its proximity. Okay, moving forward, talking about endoplasmic reticulum. So the endoplasmic reticulum is a transport network for molecules targeted for certain modifications and specific destinations, and as compared to molecules that float freely in the cytoplasm. The ER has two forms: the rough ER, which has ribosome on its surface that secrete protein into the ER, and the smooth ER, which lacks ribosome. The smooth ER plays a role in calcium sequestration and release, and also helps in synthesis of lipids. So endoplasmic reticulum, if I have the figure, wait, yeah, okay, I have this figure. Look here. This is again a representative image of an endoplasmic reticulum. Endoplasmic reticulum is directly in continuation with the nuclear envelope. It has ribosomes on it, and as I mentioned earlier, the mRNA will come that mRNA will be identified by the ribosomes. The ribosomes make the RNA sit on the this ER, and they'll translate it. Correct? For this process to occur, imagine if endoplasmic reticulum, especially rough endoplasmic reticulum, was towards the membrane side or towards the periphery of the interior periphery of the cell. So the mRNA has to travel from cytoplasm, the center of where the nucleus is situated, from that place towards the periphery of the cell. This will be a energy input work, energy expenditure work. To minimize this, what the evolution has done and how the cell is being evolved, that they have associated the rough endoplasmic reticulum just with the nuclear envelope, it's just the continuation of it. So the RNA can travel least distance it has to travel, and as soon as it travels, it comes out, it gets translated immediately. Here also certain differences are there, because every RNA, there is not only mRNA, right? You guys might be aware of it. There are different types of RNAs available. So every RNA has different lifetime. And by lifetime, I mean for how long they're stable in the system the moment they are being formed. Okay. So this is the thing, but we are not going too deep into it. I just want you guys to have a very brief information about organelles, as well as have a newer perspective in how to visualize and see cells, not just as different compartments of a cell, but as a whole. Okay. Then we have Golgi apparatus. The primary function of the Golgi apparatus is to process and package the macromolecules such as proteins and lipids that are synthesized by the cell. So Golgi apparatus, if you remember from your school days, it is also called the post office of the cell, right? If nucleus is the information center, Golgi apparatus is the post office. Why we call it post office? Because in the Golgi apparatus, things are being decided that where do we need to send this protein. There are two different types of protein. First, which are associated with the membrane. Second, which are present in the cytoplasm. So the proteins which are associated with the membrane, if you remember correctly, we discussed that there are different types of proteins which are associated with the membrane, right? It can be a peripheral membrane protein. It can be an integral membrane protein. It can be a lipid anchored protein. So all these proteins, though different name, different function, different structure, but they all are associated with the membrane. They are not interacting with or they are not present in the cytoplasm. While other proteins which are present in the cytoplasm, they won't go to the membrane. So who will decide this, that which protein needs to go to the membrane and which protein needs to stay in the cytoplasm? It is the function of Golgi body or Golgi apparatus. So if a protein needs to travel to the surface, or here by surface I mean to the cell membrane, it will go via Golgi apparatus and get towards the membrane. Correct? This is one way. The other function of Golgi apparatus is to also help in secretion. So if you know from past 2 years, if I say, we were studying about COVID-19, right? So COVID-19, different things were there, different data, a lot of data is available there. So we have vaccines for it also now, right? So if you know how a virus particle transfers itself, because it is inside the cell, it first needs to come outside the cell. Before that, it first needs to enter inside the cell, right? So all these functions which are involved, which are involving the membrane, if something is coming in endocytosis and those terminology, I don't want to introduce this, you guys are aware of it, but I don't want to go in completely different direction of discussion with respect to today especially. So vesicles just have this concept that there are certain things which are being secreted outside the cell. The best example will be endocrine system. We studied about endocrine system, right? So the hormones are being secreted outside the cell. How they are being secreted? They are being secreted in the form of vesicles. Who is making the vesicles? Who is deciding that this molecule needs to get outside the cell? It's the function of Golgi apparatus. Okay, so that is why it is called the post office of the cell, because it is deciding which things will go out and what modifications that thing has to undergo so that it can easily go outside the cell. It will not be easy for a molecule to go outside, right? Because if it is so easy that any molecule can come and go across the cell, then we don't need a semi-permeable membrane. We don't need transporters. We don't need channels. It will be a very passive process. No energy expenditure is required, and hence if this is the case, we won't be able to maintain the homeostasis of a cell. And the moment homeostasis is changed or altered, our biochemical reaction and processes will alter. That will magnify and they will show effect as disease. I hope you guys are able to connect. If a biochemical process is not at its maximum, not performing well, say if we are talking about glycolysis, if the glucose is not being broken down into acetyl coenzyme A or pyruvate and all those end product of the glycolysis, further process cannot take place. And further process will lead to energy extraction from these molecules. And if the energy extraction is not proper, you will feel tired. So as a consequence of lack of biochemical process, you are feeling tired. Hence, the homeostatic condition of a cell needs to be maintained at all the time. If you are exercising, you are running, your external environment is changing, but your internal environment is working more towards ensuring that your internal environment doesn't change. That is the homeostasis. Correct? This is the reason that Golgi body will decide which molecule to send out, which molecule to keep it in, because it is not freely available for the molecules. It is not possible for any molecule to cross the semi-permeable membrane on its own will. Okay, I hope you guys understand the importance of these things and the importance of membrane, why it is needed. This is the representation of a Golgi apparatus. As you can see, this is the nucleus, and I was saying this is the rough endoplasmic reticulum, which is closely associated with the nucleus. And those rough endoplasmic reticulum is in close proximity with the Golgi apparatus, and the Golgi apparatus will decide which molecule to send to the surface or outside the cell. Okay. Moving to the next part, our next organelle which we're going to discuss is lysosomes and peroxisomes. So lysosomes contain digestive enzymes, acid hydrolysis especially. They digest excess or worn out organelles, food particles, and engulf viruses or bacteria. Peroxisomes have enzymes that rid the cell of toxic peroxides. Lysosomes are optimally active in an acidic environment. The cell could not house these destructive enzymes if they were not contained in a membrane-bound system. Okay. So what are the major differences between lysosomes and peroxisomes? So lysosomes, they are membrane-bound sacs performing a digestive function. A similar function is associated with the peroxisomes also, that they are membrane-bound sacs performing a digestive function. Lysosomes, they contains enzyme to digest food, waste, invading bacteria and break down old organelles. While peroxisomes, enzymes in peroxisomes are oxidases that catalyze redox reactions. Lysosomes are present in animal cells only, while peroxisomes are peroxisomes are liver contains many peroxisomes to break down alcohol. Obviously, they are present in animal cell and not in plant cell lysosomes. Golgi apparatus produces lysosome, and peroxisome form by budding off from ER. It's important, right? Because right now we study about the vesicle formation by the Golgi apparatus, while peroxisomes are made from the budding off of ER. Lysosomes are associated in Tay-Sachs disease, while peroxisome are present only in animal cells. They both are like similar in one way or another, but they differ in how they are associated with the enzymes which in which their function is involved. Okay. Now talking of another organelle of a cell, it's the centrosome. So the cytoskeleton organizer or the centrosome produces the microtubules of a cell, a key component of the cytoskeleton. It directs the transport through the ER and the Golgi apparatus. Centrosomes are composed of two centrioles which lie perpendicular to each other, in which each has an organization like a cart wheel, which separate during cell division and help in the formation of mitotic spindle. I'm extremely sorry, I'm having a bad throat. Just give me one. I'm so sorry for the disturbance. Yeah. So centrosomes are composed of two centrioles which lie perpendicular to each other, in which each has an organization like a cartwheel, which separate during cell division and help in the formation of the mitotic spindle. A single centrosome is present in the animal cells. They're also found in some fungi and algae cells. So I don't think I should go too deep into it. I was figuring out certain images for centrosome, but they were either too simplistic or either they were too complex. So that's why I haven't incorporated in it. I will try to incorporate at least one representative image of centrosome before uploading this. And even if I am not able to find it, please, you guys can always go search. And if you are also not able to find it, just drop me a mail. I'll try to give some good references for this particular organelle. Okay. Next is the vacuoles. Vacuoles sequester waste products and in plant cells store water. They are often described as liquid filled spaces and are surrounded by a membrane. Some cells, most notably amoeba, have contractile vacuoles which can pump water out of the cell if there is too much water. The vacuole of plant cell and fungal cells are usually larger than those of animal cells. Vacuoles of plant cells are surrounded by a membrane which transport ion against concentration gradient. So even vacuoles, they are particularly, I am not much aware of the literature. There might be a lot of literature available for the vacuoles, but it is, I feel like that this is one kind of understudied organelle, and I feel like this has a lot of potential and we don't know much about it. So if anyone of you interested, you can always dig more about vacuoles. I'm not familiar with it, but yeah, you can always go ahead and look into it. To summarize or to get a like more detailed information about this, here I have a representative video of a basic structure of animal cell. It will give you a summarized it will give you in summarized way about the structure of a cell. As we have already covered function of the common occurring organelles, there are different organelles also, but I've covered a very limited and given you very limited information which you guys might already be having, but try to figure it out and ask different questions, right? Because the more different question you ask, the more current information which is available to you will make sense. Okay. So here is the basic structure of a biological cell. I hope you guys can hear it.
An organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Right? It was a very concise, crisp, and visually pleasing video, right? So we can always learn new things, then try to visualize it. It makes our understanding much more clear. Again, it's a best example of how the technology has advanced and how we can use those things for our own benefit. So here is one very small video with respect to prokaryotic and eukaryotic cell. So since the beginning of this lecture, I wanted you guys to have a different perspective of eukaryotic and prokaryotic cell, right? So having those things and those discussions in your mind, now look at this video from a different viewpoint and try to figure it out, that why those changes are in place. Okay, so here it goes.
On Earth, there are two major types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the simplest and most ancient types of cells. They were the only form of life on Earth for billions of years before eventually giving rise to eukaryotic cells. If you take a look at prokaryotic and eukaryotic cells side by side, you can see a number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have a simpler structure. But bear in mind that this simple structure is actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you were to look inside a prokaryotic cell, you would probably be surprised at how simple it is. For example, prokaryotic cells lack a nucleus. In fact, the name prokaryote actually gives you a hint about its structure. Kary means kernel or nucleus, and pro means before. So basically the word prokaryote means before nucleus. The inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout its cytoplasm. The cell's DNA is located in a region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as a nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or a bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess one or more flagella, which are used for movement. In contrast, eukaryotic cells contain a membrane-bound nucleus and other membrane-bound organelles. The name eukaryote also gives a hint about its structure. Eu means true, And remember that "kary" means nucleus. So in essence, the word eukaryote means true nucleus. Eukaryotic cells are larger and much more complicated than prokaryotic cells. However, they do share a few things in common with prokaryotes. Let's take a look at a Venn diagram to compare prokaryotes to eukaryotes and to also see what areas they have in common. Let's start with their commonalities. Both prokaryotes and eukaryotes have DNA as their genetic material, have ribosomes, have cytoplasm, and have a plasma membrane. Now let's look at some of the ways they differ. Prokaryotic cells are the oldest type of cell. They're small and relatively simple. Eukaryotic cells evolved from prokaryotic cells later. They're larger and much more complex internally. Prokaryotes lack both a nucleus and membrane-bound organelles. Eukaryotic cells have a nucleus and also contain organelles. Prokaryotes are single-celled organisms. Eukaryotes can be either single-celled or multicellular. And finally, the DNA of a prokaryote is usually organized as a single circular chromosome, while the DNA of a eukaryote is organized as linear chromosomes.
How to get the most free.
Yeah, so from these two videos, we have a little bit summary about a cell structure. And if you connected with our discussion from the beginning of this session, that why we wanted to study the cell in the first place for an animal physiology course, you will understand that what is the importance of knowing things which are present in the cell, cells are involved in, what can alter it, and all those, and everything is linked with each other, right? Similar things can be extrapolated for the physiology itself. As I mentioned, if the biochemical reaction is not able to take place at its maximum, it will manifest itself as a macroscopic property. So you have headache, right? For example, you have dehydration and you have headache. So your head is not dehydrated. The cells which are present in your head, which are putting pressure in your head, that is causing the headache, correct? So everything is linked, but everything comes at the end of the day comes to the cell. That is why cell is called as the functional unit of life. Everything which is being performed by the cell, this is the one which gets magnified, because we have billions and trillions of cell, gets magnified and manifests itself as the change or the movement of our being. Okay, and this is what we call as our response with respect to a stimuli. Okay, so I hope that you guys now have a holistic information and holistic point of view. Just try to adapt it. It comes with time. I'm also trying to develop it with you guys. So if you have a holistic approach, you will be able to identify problem statements, and you can actually ask some really good questions in the field of biology which are needed to be answered. Okay, with this I will take your leave as a TA for animal physiology course. It's been my pleasure delivering lectures to you, answering your doubts, answering your questions, getting your responses. Thank you so much. And if you have any query, any question at any time, always feel free to drop me a mail. You have my website link in starting of every slides which I have uploaded. You can always mail me. You can ask for like guidance, material, whichever you are struggling with. And I hope that this course might help you in your further studies, as well as helped already has helped you in attaining your goals and motives for which you have joined this course. Thank you so much. And if you have any doubt, feel free to ask. Thank you. I'm available for 5 more minutes. If you don't have any doubt, feel free to leave the meeting If you have doubt, you can ask me. I'm available for 5 more minutes. As there are no doubts, I guess I'll stop the session here. Thank you so much.